I hope I'm audible. Please let me know if there is any interferences. I would like to wish good morning to everybody. I am Sridevi Devadika and I extend a very warm welcome on the behalf of Principal, the CEO, and the management of Patkar-Varde College. On this one day, RUSA web sponsored national webinar Biofacet. And today's talk will be on impact of bioinformatic tools in biological sciences. It is organized by the Department of Microbiology, Biotechnology, and Bioinformatics. I also welcome our eminent guest speaker for the day, Dr. Susan Thomas. Hello, ma'am. Good morning and welcome. Encouragement, enthusiasm, and excellency are the major aspects of our institution, and with a similar objective of encouraging the spirit of research and innovation, Patkar-Varde College feels extremely proud to be the host for this webinar. A little about our college. The Chikitsak Samo's Patkar-Varde College was established in 1964 in Mumbai's western suburb. Since its establishment, it has constantly worked towards students and society welfare by bettering in its infrastructure and other aspects. Our college is an autonomous college that is connected to the University of Mumbai and has been re-accredited A plus grade by the NIRRCH cycle with an institutional score of 3.53. We have also received the Best College Award in 2016-17 from University of Mumbai, and we are a DBT Star College awardee. We have also obtained the ISO 9001-2015 certification. The college has grown steadily over the last 5 decades and is keeping up with the spirit of learning and growing through criticism. We have set a very high standard and we are constantly working towards achieving these goals. Our college offers a wide range of undergraduate, postgraduate, and PhD programs. In addition to the traditional program, the college also offers the emerging need based programs like we have BSc Biotechnology, Microbiology, Aviation, Computer Science, Information Technology. We have BMS, BCom with Banking, Insurance, Accounting, Finance, Advanced Accountancy. We have InCom in Banking and Finance, and Business Management. Our college has consistently bagged top positions in university and board examination. Now, to complement your learning objectives, we have with us today Dr. Susan Thomas, and it is my absolute pleasure to introduce her to you guys. Dr. Susan is a scientist E at the National Institute of Research in Reproductive and Child Health. Department of Biomedical Informatics Center. She has received a PhD from IIT Bombay and she is now a PhD guide herself for life sciences and biotechnology with the University of Mumbai. She's a member of ad hoc Board of Studies in Molecular Biology, University of Mumbai. She has a current research interest in machine learning algorithms for prediction and classification, sequence analysis, database creation, and data mining, protein-protein interaction, and disease informatics. She is an author to several and tons of publications, and she has several achievements under her belt. She has received several scholarships and fellowships. Some of the recent ones I would love to mention here. She has served Science and Technology Award for Research. She has Department of Health Research Overseas Training Fellowship. Mentioning some of her honors includes the Gargi Vishnoi Memorial Award, winner for Best PhD Thesis of Bioschool IITB 2007, and Phil Wall Travel Awardee for Protein Science Society US. Ma'am, it is an indeed pleasure to have you with us today. And before we begin this webinar, I kindly request all the participants to switch off their cameras and speakers so as to allow a smooth conductance of the webinar. If there's any technical problem during the webinar, we'll try our best to resolve them as soon as possible. Kindly post your questions in the chat box and it will be answered in the end of the session as soon as ma'am finishes her talk. It will be also open for a Q&A session. Now, without any further ado, I would request ma'am to take over the session from me and please address the webinar. Thank you so much. Susan ma'am? Susan ma'am, are you? Yes,
Yeah,
ma'am.
yes. Can you hear me? Yeah, okay. Thank you for that kind introduction. Let me just
Yes, ma'am.
share my slide. Can you see my slide?
Yes, ma'am, we can.
I hope it is in the
Yes,
presentation
yes.
mode. All right. Okay. So good morning, everyone. And thank you, Ruchita, for this invitation and also for the kind introduction that you gave me. So in this talk, I will be briefly discussing on the role of machine learning in biological research. Please understand that I'm talking about machine learning from a biologist perspective and not from a computer science perspective. So I am a biologist who has basically learned the basics of machine learning and tried to use it in my research. So in this talk, I plan to deliver it in two sections. In the first section, I'll give an overview of machine learning. Basically, what is its definition, the steps, application, challenges, and hopefully I'll be able to link you and revise you through these sections through the case studies, wherein two case studies which we have actually used in our research I would be going through so that you can understand the definition application when you see these case studies. So there are several definitions of machine learning that are available. One of my favorites is the one by Tom Mitchell, wherein machine learning is defined as a computer program which can learn from an experience E with respect to some task T and performance measure P. So if a performance of the computer for a particular task improves with experience, then we can define it as machine learning. uh, Initially it will seem a little difficult to swallow and understand. So I'll give an example to explain and understand this and appreciate this definition better. So, for example, we all use emails and we all know what are spam and non-spam emails, right? So, if we talk about this example from the machine learning point of view, what is the experience that we are talking about? Experience for the computer are the emails that are analyzed for classification. What is the task T that it has? It has to decide whether it has to mark your email as a spam or not a spam. And how would we understand the performance measure that is P? So basically it's E, T, and P. The performance measure would be accuracy. That means how many times is your email correctly marked as spam or not a spam? Now it always comes with a cost. Like I don't know how many, whether you have statistics and sensitivity, specificity, all these terms quite well understood and appreciated. Uh, when we want to increase the sensitivity, sometimes the specificity goes down. So for me as a user, I would rather like suppose someone tells me that what, it's not possible for me to give you 100% accuracy, but I will try and ensure that your specificity, that means your false positives are low, or your sensitivity, that is false negatives are low. I cannot do both together. I would rather say that I'm okay with one or two non-important emails coming as in my primary folder, but I would not like to have any important mail going into my spam folder because that's a huge loss for me. But we all know that as of today, we still see that not many, I would say, few of and our important emails are automatically marked as spam and put into the spam folder. So many times, especially from journals, when I send my manuscripts and I'm wondering, where is that email that they have sent? I do not see it in my mailbox. Then, when I look into the spam, by mistake, Gmail has actually predicted it as spam and put it into my spam folder. There are several reasons for this. I'm not getting into that, but what I'm trying to tell you is that this is some kind of an AI or an ML, wherein the algorithm is trying to increase its performance based on analyzing emails over a period of time. And the task, as I mentioned, is to decide whether any email is a spam or not. So when it comes to the learning process, there are several steps from the user or the researcher point of view. The first step is to define your problem. That is the task. So for a researcher, if you're interested in machine learning, you have to be very clear about what task do you want the computer or the software to perform. Once you have identified the task, automatically the next step is your experience, the experience you're giving to the computer. That would be the data collection. Once the data is collected based, completely synced with the task, next is to process the data. That is pre-processing the data, which I will explain in detail in the coming slides. After that, you will have to train the model, then validate the model, that is the performance of the model, and finally we'll have to deploy or use it in the real world. So let's let's see each step by step. So the first is the problem definition and selection of the study design. So as I mentioned earlier, we have to precisely define the problem. We'll have to define the goal for performance. Like I said, uh, what is the performance that is acceptable? That all depends on whether a similar algorithm already exists in the current scenario. For example, if I have to come up with a ML algorithm for, say, weather prediction, we all know that there are several algorithms for predicting the weather. So if I have to come up with an algorithm, that would be my benchmark. Is it better than the existing prediction algorithm? In case there is no prediction algorithm currently available, then you do not have an algorithm to benchmark it against. Then probably anything between 60 to 70% accuracy would still be acceptable. And the third important step is to select the appropriate study design. Let me explain this with an example. If you want to develop a diagnostic algorithm for a disease, so for example, let me say if I want to make a ML algorithm for, say, predicting psychological distress among individuals, then the data that I should be using for training my algorithm or for the experience should come from a case control or a cross-sectional study where there are clear data points of healthy and diseased individuals at a particular point of time. I hope I have made myself clear. I'll repeat it. So I hope you understand what is case and control. Case and control is we have the kind of features. And when I say features, these are basically the parameters of a person who is having psychological stress, maybe the diet, physical activity, the emotions that the person feels through. Each of them as a vector would be called as features. And if I have to make an algorithm for this, I will have to collect data from such case control cross-sectional study so that I can make a meaningful algorithm. However, if I have to make an algorithm for, say, disease risk prediction, that is uh, like, for example, what is the risk of a person who is chewing tobacco to, say, get oral cancer? So naturally, we all know that a person who takes tobacco today may not end up with cancer tomorrow. But then you'll have to kind of look at the data of these individuals over a longitudinal period as a cohort and understand what are the markers for individuals who end up with oral cancer and who chew tobacco, whereas for individuals who do not end up with cancer but yet have a good amount of tobacco consumption. So depending on what is your goal, you will have to select your appropriate study design. Next, a very important aspect is the experience or the data collection. Now, as I said, based on your objective, you will identify the source of data. Many times, the source of data will either be using the existing data that people have reported in literature. uh, like, and I will talk about the sources of data in literature or in online resources. But you could also generate new data, wherein you could probably do a statistical kind of sampling and then ask through a questionnaire. But then remember, for ML, more the data, the better. So the source of data could either be new or you could use existing. But very important is to define the inclusion and the exclusion criteria. So I'll explain this again through an example. So as I said, there are many publicly available data sources. NCBI is a good, kind of, one point center where you could get data from literature, say from PubMed, the Gene Expression Omnibus, Geo database, Genome. So many, data sets and databases are housed in NCBI. So each of these, each of these web sources are an excellent source of data. But you have to be very careful as a researcher to identify which data you want to pick up from these resources. I'll give you an example. Suppose I want to understand what are the gene expression patterns which are important, say, for uh, individuals with PCOS versus endometrial cancer. In the sense, what is the likelihood of PCOS and endometrial cancer gene expression status being similar or dissimilar? I cannot just go to the GEO database and say, give me all studies of PCOS, give me all studies of endometrial cancer. Remember that the data that has been submitted by the researchers into these databases all are based on what was their task or their goal. So when we are doing a secondary analysis for machine learning, it is very important to manually curate all these databases to fish out data which is important for the analysis and to exclude which will actually create a bias. Like for example, there would be data on PCOS gene expression where the individuals are on some kind of treatment. There would be data where individuals are not on any treatment. There would be data where there would be individuals on PCOS with, say, hypertension. So when we take the gene expression of all these individuals together and say, what is the difference? Can I predict what is the risk? There is a lot of noise in the data that you have put in, which will be very difficult for the algorithm to figure out that this data is not actually correct for analysis. So the correctness of data for analysis is still largely manual intervention. So that is where humans come in, where we say that you are not supposed to see this data, and this is the data that is going to be seen for this particular task. And believe me, that is a huge amount of data, a huge amount of work with respect to data curation. The next step for ML algorithm is the data pre-processing part. So the data that you get from the online resources or through, say, a or questionnaire uh, need to be pre-processed. The first step is to see how clean is your data. So what do I mean by ensuring that the data is clean? So you have to check for the quality of the data and the completeness of data. Many times, if you're looking at, say, health records from hospitals, or we are collaborating with clinicians, many times the data analysis is through clinical collaborations. So the clinicians would give the health records of individuals, and we would see that many of the data points are actually not complete. Uh, for example, if I want to make an ML algorithm and my task is to predict what is a successful delivery outcome in a diabetic mother, can I predict which of the diabetic mothers will end up with a successful delivery without any complications? Whereas, can I predict mothers who are diabetic who would probably need some kind of treatment or intervention later on? So, the initial experience that I give the algorithm would be the biochemical parameters of mothers who are diabetic, and then I would also would want to know, like for example, what would be the features? The features would be what was the diet of the mother just before pregnancy, what is the diet followed by the mother during pregnancy, what is her physical activity, what are the medications that she's on, what are the comorbidities or the other diseases that she's currently facing. So these are important data points or features which will help the algorithm to learn what to predict that which of the diabetic mother, given these parameters, would end up with a successful delivery or will have need for some kind of a treatment intervention. So now when I look at, imagine you're seeing this data I want you all to visualize an Excel file where one column is, where outcome, successful delivery, second is not successful or need treatment intervention. So when I look at the data points wherein it shows you what was the diet before the mother had, before the lady became pregnant, did she have her folic acids, her vitamins, all those data has to be complete. It is possible that some of the ladies would remember and say correct information. Some of the ladies might, give wrong information just because the person has forgotten. Some of the people would not even remember and say that I do not know. Especially it comes very important is the second and the third trimester, where you want to know the blood parameters in the second and the third trimester to know what is the outcome in delivery. If the patient does not follow up, would not come for the second and the third trimester and suddenly come only for the delivery, many of the important crucial data points are missing. So when you want to make an ML algorithm, you will see that you will find an Excel sheet with so many erroneous information, so many information which is actually missing, and it will be very difficult for you to, kind of clean that and make it into a form which is useful for ML. And I'm not saying this as a one in, a million situation. Majority of the time, this is the data that we get, which is incomplete, which has a lot of errors. Because remember, there human error is possible even in coding, writing the data, and recall. A person's recall to past events might not be really great. So all these affects the quality of the data. Now, next coming to what could you do with this missing data? Why? Well, we can do many things. One is we either remove them. We can remove if we have a good number of complete data. Then the best thing to do is to remove the incomplete, rows. Suppose our data is already less. Maybe I have only 100 individuals who are enrolled in the study, and I need those 100 individuals minimum to get a good performance. So what I can do is I can do something called as imputation, wherein I look at the mean of that particular column and then just put a mean value because I cannot take a missing value in my algorithm. Or I can just put a constant value, say like 100 or something, to say that for any missing row, I just put a random value of 100 because I know that this is actually missing and the computer can still take it. But but incomplete data definitely comes with a cost in the performance. Next very important aspect, especially when you're dealing with biological research, is outliers. So as the name suggests, these are outliers, meaning they lie outside the normal range. Now, if you let me use a pointer. Now, if you see this particular point, we have x-axis as age in months and y-axis as the weight. You see that there is an individual uh, whose whose weight is much higher than what is generally accepted in the range. This can be visualized either as a scatter plot or as a box and whisker plot, as you can see, where the interquartile ranges are depicted. There is an outlier here. So what does an outlier imply? So the outlier can imply that they are actually present, or it can be an erroneous way of writing the data. Like for example, in ICU or an NICU, where the children's ward, a data entry person would have by mistake put a zero. So of three, the person would have put 30. So that is an erroneous data, and when you plot it, it looks like an outlier. Or there could be a genuine case of a high weight because of a pituitary tumor or some kind of growth anomaly. So it is very difficult for the analysis person to figure out whether it is actually an anomaly in writing or an error in writing, or actually there is some kind of a biological issue for which the outlier is there. Since many of you all are students who are attending this workshop, I'll give you another example which you can understand better. One is suppose I want to make an ML algorithm to decide whether what is the correlation between number of hours spent in your college, Patkar College, versus the marks that you get in your exam. By now, we know that we are supposed to expect a positive correlation. That is, more the number of hours you spend in the college, attend the lectures, you're expected or predicted to get a better result. It is possible that, when you plot this as a graph, like for example, the x-axis is number of hours spent in the college versus y-axis is your performance as marks, you will see that some points will be there where number of hours is less, but the performance is very high. So if the computer has to analyze this data, it will get confused. It's like, what is it? Is it a positive correlation or is it a negative correlation? So the outliers kind of confused with respect to the relationship between the variables. And and again, the reason could be probably, I'm just giving a hypothetical example. It could be that there is a cluster of individuals who probably are very intelligent, or they may be going to some kind of coaching class. They may not be attending the lectures, but they will be going for tuition centers and they'll be learning there. So we cannot actually use these outliers in the analysis, and it is better to remove them because they would create unwanted or wrong predictions. So it is important to detect the outliers and decide on their inclusion or exclusion, as these outliers can bias the decision process. It could, as I mentioned, it could be an error in the data capture or an anomaly. So the researcher has to look into each of it's not very easy. You see an outlier, you remove it. It's not as simple as that. You need to understand why the outlier is there in the first place and then take a informed decision that then what should I do with the outlier? And especially, as I mentioned, in biological research, outliers are very, very important, and we constantly see them because individual organisms have high level of heterogeneity, and it is bound to happen. Next, very, very important aspect is to reduce the data or the dimension reduction. Why we say that the number of data points are very important? We do not want the features of the data points to be much more than the number of data itself. So when I say features, suppose we have 100 individuals who have enrolled in the study, we do not want, 1,000 parameters to be captured for each of the individual. So that kind of becomes like a high dimensionality curse. So some of the features, what we capture may be redundant or highly correlated. So again, let me give you an example. If I want to understand what is the importance of socioeconomic parameters on, say, survival, I would be Probably in my questionnaire, I'll be asking questions like, where do you stay? What is the size of your house? How many bedrooms do you have? How many cars do you possess? What is the model of your car? And so many such data. What is your salary? So each of them, you can imagine them as a column in an Excel file. So don't you think that all these different parameters, that is, the number of cars you have, the size of your house, the income, they're all kind of correlated, and they could actually be just clubbed into one variable rather than having many different types of variables. So this is one thing what I mean by redundant or highly correlated. Second is that second issue with high level of data feature is that redundant and irrelevant features, they reduce the interpretability of the model and lower the performance. Let me explain this. Again, with the same example of what I said before, suppose the task is to understand the socioeconomic parameters and survival of, say, ovarian cancer, women with ovarian cancer. And if I have taken all these data points for the socioeconomic, because we do know that socioeconomic parameters do play a role in the treatment that they get, in the kind of lifestyle that they have, in the type of opportunities that they do get with clinical trials, so on and so forth. But if I have to put data like, for example, number of cars, or do you drive a car? Yes, no. And suppose for many of the data points, I get no. Is it right to say that, when I make a model? So remember, when I make a model, at the end of it, the biologist is also interested to know what is the correlation between the various parameters so that we can do some kind of therapeutic intervention to that. So if my model says that, if the individual has high income but does not drive a car, the the probability of survival is higher. Now, not driving a car could possibly be because the lady does not know how to drive the car. As simple as that. It has nothing to do with socioeconomic status or the survival. I'm not talking about the survival because of road accidents here. Then, of course, it makes a difference. But here I'm talking about, since a lady does not know how to drive the car, but her socioeconomic status is high, you can always club her as a data point for high socioeconomic status. So unnecessary parameters into your model can make it very fuzzy and very difficult to interpret. And very important other reason is that high number of dimensions of features can increase your computational time, which is not required. So the onus of data reduction is to obtain a reduced representation of data volume, which can achieve almost the same results. So remember, we do not want to reduce the performance. If by deleting a particular column, the performance gets drastically reduced, then we would like to retain it. So it's not that you keep reducing it, even if your performance goes down, so that you can have a small bunch of data. No. And how do we do this? So feature reduction can actually be part of your ML itself, as I will be showing you in the subsequent slides. Next aspect is transformation of data. So when I say transformation of data is when you are replacing the variable by a function of the same variable. Uh, so common data transformations are scaling, decomposition, and aggregation. Let me talk about scaling. So scaling is, I hope you can see this column. I hope it's not too small for you to read, where I have a column called age and then a salary. So if I look at the range that is shown for age, uh, it is it is much lower than the salary range. So if I have to normalize all my different features, so here age is a feature, salary is a feature. If I have to normalize the range to 0 to 1, I can use something called as a min-max normalization, where you can see this is the formula that is used, wherein each of my columns will now have the same range of 0 to 1. So this is what is meant by scaling transformation or normalization. Next is decomposition. Decomposition is when a data can be split into the constituent parts so that it can be made more meaningful. Again, as you can see here, the date represented here can be decomposed to a day, month, year. Probably this can even be, kind of superposed into the season to understand what is the influence of season on diseases and things like that. So this is called as decomposition. Next is aggregation, where features can be combined into single feature, which are more meaningful than when they are separated. For example, suppose I have individuals whose age, gender, height, weight are given. A better way to remove height and weight is to actually put a body mass index, which is more meaningful because we all know that more the height, more the weight. But what we want to know is that what is the body mass index to understand if the person is obese or not. And body mass index, by definition, is a person's weight in kilograms divided by square of the height in meters, and which is much more a true predictor of a disease than compared to a height or weight. So this is what is called as aggregation. So these are the different steps that are involved before we actually show the data or start, modeling the data. So we do a scaling, we decompose, we aggregate, we remove the outliers, we clean the data. If you have to if you have to change the missing data using imputation, we do that. After all these steps, only we start to the next level, which is we train the model. So once the data is perfect to the extent that we can do, we give this data for training. So here, what are the steps involved? So the steps involved are basically we take the entire data and but that is, for example, if I have data of say 100 women with diabetes and pregnancy outcome, what I would do is I would split it into two sets randomly without, giving any user bias. One is where I train the model, where I ask the model to experience what is correct or no. And then I will use a holdout set or a test data set to see whether my model, how does it perform on this test data set. So the data can be, so I have an original data set of 100%. I randomly split it into training and test data set. Always the training data set is higher than the test because that is the experience the algorithm gets to learn. So you cannot keep it lower. The more, the better. But you do not want to end up with a model which is very good with the training, which will perform very poorly in an unseen set. So hence it is very important to have an unseen test data set. And from this training data set, it will learn using the different features and finally come up with a predictive model. And on this predictive model, I will run a test data set to show that, okay, my model is not only good with the training set, it also performs well with an external test data set. And how do we do this performance prediction? Is one step is to cross validation, where what we do, there are two methods. One is a k-fold cross validation, and second is a leave or not cross validation. What is a k-fold cross validation? So in your training data set, what we do is we kind of split it into five unique, mutually exclusive data sets, and we ask the computer to learn from four of the folds. Suppose it's a five-fold. From the four folds, it will learn and will predict the fold that is left out. So as you can see here, in the first split, the fold one is not used for training. The fold 2, 3, 4, and 5, the computer learns from these, and then it will predict what is the result on fold 1. Similarly, the second split, the fold 2 is not analyzed. In the third split, fold 3 is not analyzed. And finally, it is pooled to understand how good is the prediction accuracy. While in the leave one out cross validation, every record gets a chance to be tested, meaning every record or every data point is not used for analysis while the others are. And finally, you take an average for the n different training data sets. So these are the two types commonly used for cross validation. Now coming to the most important aspect, which is the validation of the model, which is also called as a performance, as I mentioned. So once we develop a classification model, it has to be validated. And the confusion matrix is a table which is most often used to describe the performance of the model or the classifier. Remember, the algorithm can either be a classification where you want to, classify it into different data sets like diabetes, hypertension individuals, or it could be something like to predict a treatment outcome with respect to, a continuous variable. In that case, it becomes a regression model. I'm not getting into the details of those, but remember that a confusion matrix is how a performance of the ML is usually reported. Now, to explain this, let me take a very arbitrary example. This is quite usually seen in the web, where they use it to show true negatives and false positives. So, for example, if you have an ML, just very arbitrary, of course, ML does not work on size of belly for predicting pregnancy. But imagine if you have an ML algorithm which looks at the belly circumference and then suggests whether you're pregnant or no. A man being just because he has a huge belly and being said that you are not pregnant would be a true negative. That means you are saying that the outcome is not pregnant, hence it is negative, and it is a true negative. While a man with a huge belly, because the belly is high and the ML says that, oh, you're pregnant, that becomes a false positive. It's also something called as a type 1 error, where we reject a true null hypothesis and it is wrong. So the null hypothesis was that the individual is not pregnant, but the ML algorithm rejects it. So it is a false positive and it increases type 1 error. Now, next is the false negative, wherein a lady who is pregnant, but the tummy is not, huge. The algorithm predicts as not pregnant. Again, it's a false. It's not a negative. It should have been positive. So it's a false negative. And false negative is a type 2 error where we accept a false null hypothesis. And true positive is, of course, the belly is big, and ML algorithm says that yes, the individual is pregnant, and it becomes a true positive. So a confusion matrix uses these different true positive, false, negative in different formulae and figures out the sensitivity, the specificity, the accuracy, what is a positive predicted value, what is a negative predicted value, so on and so forth. And this is determines the performance of the ML algorithm. Sometimes it is very important that the sensitivity is high and not the specificity. Like I said about the spam emails. So it depends on what is important. Whether your true negatives are important or your true positives are important, you can give more emphasis on the positive predicted value or the negative predicted value. Earlier, we would just go by the accuracy, but that is not correct. We have to report these different sensitivity, specificity, positive predicted values to finally understand how well your ML algorithm performs. And coming to the types of machine learning algorithms, we basically have two, and there are many others, but mostly they can be classified as supervised and unsupervised learning. We also have semi-supervised learning, reinforcement learning. I'm not getting into that here. So when it comes to supervised learning, the difference is that we already have a labeled class. Like for example, if you see this example, we for the machine we introduce the data as this is a rectangle, this is a circle, this is a triangle, this is a hexagon, and then we give a lot of features of these. Like for example, it could be it could be the diagonal length, it could be the side parameters, so on and so forth, because you cannot just give a shape, it has to be features. And then the machine makes a model, and when you give a test data of these same labels, it will correctly or not correctly predict the label for the test data set. So this is supervised learning. Here the thing is, we initially itself we will have to label the class. We're going to talk about, either it could be, these are apples and these are strawberries. So if I give a fruit, classify whether it's an apple or a strawberry. So since we're giving labels earlier itself, it becomes a supervised learning. Unsupervised learning is also possible, where we give a list of different features. Again, you can think of it like an Excel file where there is no outcome variable, but it has several different features, and then the machine will learn it, and the machine will segregate it into different clusters. It's basically a clustering algorithm, and then from the results, you can sometimes make very interesting observations. I can give an example for unsupervised learning. It could be like, for example, if I take the blood sample of COVID positive individuals. So I have a sample of say, some 10 hospitals. I take the blood profile, including the immune related parameters, and I give it to the machine, and the machine does a clustering and kind of gives some 5, 6 different types of clusters. And in these clusters, since there are no label, I can only see is cluster 1, cluster 2, cluster 3, cluster 4. Now, in these clusters, when I go back and map these individuals, it will be very interesting to know if the vaccinated individuals of this COVID positive have a different profile of their blood parameters and immune parameters as compared to the non-vaccinated. Or, for example, one who has had 2 vaccination doses versus 3 vaccination doses, are they clustering differently? So, both supervised and unsupervised learning have their own advantages and is very important in biological research. All right. So I've quickly taken you through the different parameters of steps of machine learning and important terminologies. I will quickly, in the next few minutes, explain 2 case studies that we did in our lab and explain how we use machine learning in our biological research. So the first case study is we wanted to predict antimicrobial peptides. So what are antimicrobial peptides? Antimicrobial peptides are basically peptides which can actually kill uh, a microbe. It could be antifungal, it could be antibacterial, it could be antiviral, it could be both antifungal and antibacterial sometimes. So we wanted to predict, given a sequence, can I predict that the sequence is going to be an antimicrobial peptide or no? So this was my task. This was my goal. And coming to the benchmarking, that is, as I said, when I start a prediction algorithm, I should understand are there any prediction algorithms already existing in the market? And if I have to come up with something today, it better be better than the existing ones. So it turns out that there are several different prediction algorithms for antimicrobial peptides. So we took one of two of them to show that how good is our algorithm as compared to the existing. So what is now the study design? The study design, as I've mentioned, is you will have to also think about whether you're going to take data, whether you're actually going to make a structure activity relationship data where I'm going to synthesize several different peptides, use their structural parameters, and then say whether these peptides are antimicrobial or no, or I could use the existing online data, which I mentioned earlier. So we went with the approach of using existing data that is already reported by literature, by researchers in literature. So what we did is we got information of experimentally validated antimicrobial peptides with information on sequence, what was the antimicrobial activity, what is their origin, is it a natural origin or was it synthetic, and other various different parameters from the literature. So we read papers, we extracted this information, and we created an Excel sheet. So this is as simple as it can. I can say it, but it is not as simple as it sounds. So there are two different types of peptides, which I said natural is the source is natural. Synthetic is what is designed by de novo. That is, it is not, it may not have the natural amino acids. So we use it separately because we knew that we should not be clubbing these two together. Now, there's something called as positive and negative data set. Like, for example, in that example that I told you about, we have spam and not spam. So similarly, we have something here which is antimicrobial. We also have something which is not antimicrobial. So the algorithm has to decide whether if I give it a sequence, should we classify it as a positive, that is an antimicrobial peptide, or should I classify it as a non-antimicrobial peptide? Now, here I want you to listen to me very carefully, is that such kind of data sets, which is called negative data set in biology, is very, very difficult, unfortunately, because people do not report that this sequence is not antimicrobial because they would not either have information, or even if they have information, no journal would probably take a data that, you know what, I synthesize this peptide, I check for antimicrobial activity, and it is not antimicrobial. While researchers like me who are creating ML algorithms will find it very useful, no journal will accept such kind of data. But now we are, I mean, the scientific world is increasingly appreciating the importance of negative data, and there are journals which kind of encourage negative data, but you cannot expect it to have a high impact factor and impact factor at all. But but this is very, very important when it comes to ML algorithms. For biological research, negative data set that is not antimicrobial in this case is very, very difficult to find. So what we did is, since we know that antimicrobial peptides are generally secreted in nature because it has to go and attack a microbe, we looked at all those peptides in UniProt which were not secretory in nature. That is, they are not secreted, or they did not have an annotation in UniProt as antimicrobial, which is not absolutely correct, because just because it does not have an annotation as antimicrobial, you would still not be 100% sure that it is not antimicrobial because a person has not done a antimicrobial assay on it. So what I'm trying to again reiterate is that the negative data set is a reason why we may not be getting very good prediction accuracies. So we looked into PubMed, we went into NCBI protein database, we went into PDB, we curated this literature, and then finally, with information on the sequence, activity, and source of origin, we created an Excel file. Usually this kind of work, and especially for this paper, I had a very good assistance from Khalsa College MSc Bioinformatics students. So they really helped me along with our staff, because it's a lot of data that they had to read and extract. So one thing that was told to me finally is that while they also got authorships in the paper that we published, one of the issues is that when they go and submit their thesis, the external examiner will ask that, oh, that's what you did? You just read papers, or you just extracted information? It's very sad that even today, researchers do not understand the importance of students knowing how to read literature, how to interpret literature, and how to extract information, because someone has to do this, and it is not a trivial task. So when anyone does anything like data curation, please do not take it as a very simple task, because the individual has to read a paper and understand the scientific way of writing and extract information. So this is very, very important. I just wanted to make a point of this. And next is the data pre-processing, as I mentioned. So here we wanted to exclude sequences which are not standard amino acids, where the length is greater than 100 residues, where the similarity is very high, because we didn't want redundant sequences. And with this approach, we could get around almost around 2,000 sequences which were positive, and 4,000 which were negative, which was basically random sequences that we picked up from UniProt. And here comes the question of data reduction. So I know you all will understand that a sequence is like a string where there's a string of different amino acids or residues. So obviously we'll not be putting this into the algorithm. We'll have to convert it into vectors, which could be as again an Excel file with the length of the peptide, what is the net charge, what is the hydrophobicity index, what is the helical propensity, what are the dipeptide composition, so on and so forth. There are many online softwares which give, like for example, Profeed server gives you this vector from a sequence. And so we kind of calculated 275 features from these peptides. And what we did is at every step, we reduced it by 50% using ML to see what is the influence or the impact of feature reduction on the performance. And finally, we realized that 64 features was the best to give an optimal performance, and we used 64 features. So again, what we did is we divided it into training test and an external test. Why I kept an external test is also because I wanted to keep a data set which is not used by the benchmarking algorithm. So if I have to compare whether my algorithm works well with the existing algorithm, I have to take care that the set that I use, I have not used for my training data set, nor has the benchmarking algorithm used it in the training data set. So basically, all the peptides which were reported post, very recently was used as an external test data set because I know that these sequences would not have come into the training of the benchmarked algorithms as well. And we found that SPM, random forests, and artificial neural networks were very good, and especially RF did very well. So DBASSP, as I mentioned, was one of the benchmarking algorithms that we used, and we found that ours is called as CAM, just collection of antimicrobial peptide database, and we found that our accuracy was more or less good. And finally, coming to the use for the public, so we have created this as an online resource, and anyone can actually use it freely in the CAM database. It is one of the highly cited databases for antimicrobial peptides. We have more than 1,000 citations for this. So, and it has been recently again the updated version. So this is the fourth version that we have published. All the four versions are published in Nucleic Acid Research, and the latest is in the 2023 database release of Nucleic Acid Research. Another, if time permits, can I have some time here?
Yes.
It's already, I think, around 40. Yeah, all right. So another case study that I would like to explain is predicting the cause of death in verbal autopsy. I don't know how many of y'all have heard about verbal autopsy. So verbal autopsy is basically it helps to determine the probable cause of death where there is no medical record. So in India, especially and in the rural places, birth certificates are important if a child has to, kind of enroll in a class for in schools. But there is no incentive for a death certificate. So what happens is many times we do not have any certificates to understand what was the cause of death. And in a country, especially developing countries, we need to know how many lives are we losing to what kind of illnesses so the government can take appropriate policies. So we have something called as the World Health Organization as kind of also brought in this concept of using verbal autopsy to kind of fill in the gap of the death records. So if there's a million death study that has happened in India where non-medical, that is graduates or below graduates, field workers, they were trained to record written narratives from families or other informants in the local language. So they go to the rural places across India and they would be asking the relatives that was there a death that happened in the family, what was the symptoms, did that person have cough, cold, did the person have diarrhea, in their language in the way they understand. And these records were then scanned and randomly sent to two of around 130 trained physicians. And then the physicians could see these scanned records and prescribe or ascribe a cause of death to each case. So the physician's keywords were basically the symptoms that the physicians would pick up from the narrative of these these field workers. So the question that we asked is that can we predict the cause of death if we only had these physicians keywords? If we could, what is the implication is that if we could create a more structured verbal autopsy tool saying that we just have to capture these particular keywords and our cause of death could be easily predicted. So what we did is we took the data of around 18,826 unique childhood deaths that have happened in India at the ages of 1 to 59 months during the time period of 2004 to 2013 from the million death study. 70% of the death was caused by six infectious diseases. And we use the physician's keywords. And like, for example, this is like the Excel sheet that you can think of. Like we have a cause of death. And then, of course, when the fever was present, we code it as binary. So one, no fever, zero, cough, vomiting. So like this, around 35 columns were populated. And then we did something called as a we wanted to know whether this makes sense with respect to the disease point of view. So we clustered this using hierarchical clustering. This is the example that I spoke about of non-supervised learning, where we are not giving any labels. We are just putting this particular, without this cause of death column, we just gave everything else. So we did not put the cause of death into the clustering. That is a label that we are using. So if you give this label, it becomes supervised. If you remove cause of death, it becomes non-supervised. So removing the cause of death parameter, we went around doing the hierarchical clustering, and we said ask algorithm to make six clusters, and it actually correctly gave six clusters. How do we know it's correct? Because when you look at the different clusters that it has generated, I see one cluster as rash and abscess, which actually talks about a measles cause of death. Then there's something called fever, chills, fever, and high fever, which is actually malaria. And then there was another color-coded cluster where it is there's cough and fast breath and wheezing, which actually spoke about the pneumonia cases. So this shows that the physician's keywords were actually could kind of cluster them into different groups as expected, even though we did not give the cause of death in this particular analysis. And finally, we did a supervised learning, giving the cause of death as an outcome and having an 80-20 training test data set, and we could get very good prediction accuracy for most of the diseases. And this work is now published in PMC Public Health. So yeah, so that's about the second case study. So I gave two examples where very different from each other, where one we have the supervised and non-supervised from basically health records and narratives. And second is where we have used protein sequences for machine learning. So quickly, what are the challenges that are faced by biologists is one is the data. So if you all have to do some machine learning on biology, it's very, very difficult today because most of the data is not shared. The valuable data that we have is not shared. The high quality quantity data, especially negative data set, as I spoke about for antimicrobial peptides, is not available. There's a lot of noisy and missing data that comes from the clinicians, which we have to work on. Interpretability is a very important issue with biological data because it's very difficult to interpret the models. And basically, when you have different, like for example, different genes are important, we should be able to interpret why these genes are important in a particular disease. And sometimes it's very difficult to give that interpretation. And we usually, if you have to, since the data is small and we make a model on a small level data set, finally, when you have to validate it in a population with higher heterogeneity, the ML algorithm kind of fails. That's because it has not seen that kind of variance in your model. So that is another challenge. And at the end of it, one solution for all this is, getting high quality and quantity of data. Finally, I would like to acknowledge the funding agencies, basically ICMR, as I'm from the ICMR Institute. Our center is also a DBT funded Center for Advanced Research in Women and Child Health. And I'm very proud also to share that much of the slides that I have created here today are basically the help from Ms. Ulka Gavde. She's my PhD student and a project associate. And also happy to share that she's an alumnus of your college. She has done her BSc Biotechnology and MSc Bioinformatics from Patkar. So, and she's also a co-author in maybe I think a first author in both of these publications as well. So a big thanks to Ulka. Thank you. Yeah, that's it. I do not have anything else to present, and I'm open for questions.
Thank you so much, ma'am, for enlightening us with the role of machine learning in biological research. And I'm sure that all the participants have got a fair knowledge about importance of machine learning. We now move on to the question and answer session. Now, I would request our participants to post questions in the chat box. Okay, we have our first question, ma'am. Um, which one is the best machine learning algorithm for beginners?
Uh, all right. So actually, when you ask which is the best machine learning algorithm for beginners, uh, I would rather say best for beginners from the learning point of view, then it is all these SVMs, RFs, VRR packages. You could, use tutorials to learn them. So SVM, RF, ANN, there R has made life very simple for biologists. So I would suggest to look at these R packages and learn these kind of machine learning algorithms. But are they the most important for understanding and getting the right performance? May not be. Sometimes they may be. Sometimes simple ML algorithms may give you as good a result as more complicated ones. But that is for you to see by actually doing it.
And we have another question. What is the accuracy of machine learning models? Can we trust the predictions?
What is so generally the researcher who creates a machine learning algorithm will put out their confusion matrix or the performance measure. So it's up to us to understand that, and then please understand that the performance measure is based on the sample or the test data set that they have used. So if that is not a true representation of the population, you might not get a good outcome. But it's always good to see the confusion matrix and understand what is the performance of IML algorithm. And it also depends on the data available. So in some cases, 70-80% might be good because the data is not really there. But in some cases, you need something like 80-90%. It depends on the question that you're asking.
We have another question in the chat box. Any research article or paper you would recommend?
Oh, several. Um, I would also, I would really start you can you can actually go through the R packages and the tutorials because they are really very good the way they explain. And to understand machine learning per se, you can also see there is something called towards data science. And there are a couple of them which I can probably. Ulka, are you hearing me? Can you put them in the chat box, please?
Yes, ma'am. I'll share the link with here in
Ye-
chat box.
Yes, please do that. So Ulka will share the ones that we have found very useful, and you all could use the same.
Okay, ma'am. And we have another question. Any tools online or offline that can be used to reduce data, that is data cleanup or data pre-processing?
uh, to clean the data. So generally cleaning data, that is where, as I said, human intervention is very important because you need to understand how why there is a missing data, how to whether you should use what kind of imputation. You should not be doing it in an automated manner. So I is that the question that was posed? Like how do we do data cleaning?
Yes, ma'am.
Please can you repeat the question again?
Um, any tools online or offline that can be used to reduce data pre-processing, like cleaning of data or outlier detection?
See, like for example, the scaling transformation, you need to understand, you need to really understand the concept of your columns. You cannot just blindly give it to a data server and say that do you do the cleaning for me. That is not correct. But things like as I said, min max scaling or normalization is just a formula. So those can be easily done. But others, you have to have some idea about what columns are you dealing with. As I mentioned, why are you finding the outliers? You need to have that level of manual intervention.
And we have one more question.
I think
Uh,
Ulka has put up the different links on the chat box. Please, I request the
yes,
participants,
thank you, ma'am.
please use that.
Uh,
Yeah.
what is the most common issue when using machine learning?
Most common issue in machine learning is, as I said, the data itself. Once you have clean, good data, there is no problem. So the data with respect to the sample. So sometimes what happens is that you have data, like for example, I will give an example of antimicrobial peptides itself. So antimicrobial peptides come from different families of proteins. So if I have taken only one type of family, say the frog antimicrobial peptide family, the features will be very restricted to the frog antimicrobial peptide. So and even though it will give me a good, and if I'm using a test data set again from the frog family, I will get a very good performance accuracy, but if someone wants to use it for some other families, it will be very poor. So the task has to be very clear. The performance matrix has to be well appreciated, and then how you use an algorithm developed by someone else should be based on what was the task that person has developed it for. So if I'm using it only for protein antimicrobial for frog antimicrobial peptide family, you should not generalize it for antimicrobial peptides. And second, as I'm repeating for the that data. That is the most important limitation that we have.
And one more question we have here.
Yeah, and one more thing is, there's a lot of predicted data that is available in online resources, like predicted function and all. We are not supposed to use predicted annotations for machine learning because that way we will be running prediction on already a predicted, state. So always use experimentally validated, well annotated data for your machine learning. Especially for bioinformatics students, whenever you're planning to do something, do not take any predicted hypothetical postulates and then start using that for running another prediction model.
We have one more
Yes.
question. How much difference is shown when we predict phylogenetics using maximum parsimony or minimum evolutionary methods compared with machine learning?
Um, that is a very difficult question to ask because everything depends on a case-to-case basis. That is why we have multiple algorithms. Like for example, if we have even within ML algorithms, we have SVM, we have RF, we have ANN. We will not just say that SVM is very good, so I will use SVM everywhere. Case-to-case basis, it depends on whether which kind of an algorithm works well. Hence, whenever we have these prediction algorithms, we actually run it on multiple types of algorithms if there are multiple algorithms catering to the same task. So if the task is say predicts evolution based on something, the same task, if you have different types of algorithms, my recommendation is please use everything because there is no predefined rule saying that this algorithm is always the best.
And one last question. How does machine learning help drug discovery?
How does machine learning help drug discovery? So again, drug discovery is a very big topic, very, very vast topic. So what part of drug discovery are you talking about is an important question. For example, the gene expression data sets that you saw. So the gene expression data sets could be used as to identify a target for drug. Now, if I know that from the gene expression data sets for, say, cancer versus non-cancer of a particular type, I cannot very broadly generalize cancers, but for a particular disease, if I see that, yes, certain genes are actually overexpressed or underexpressed using my ML algorithms, I can see whether those could probably be targeted. So in that way, it can help in drug discovery. I can look at what are the possible mutations that have led to drug resistance and then say that, any feature or any domain motif in this particular structure highly likely to, be part of the resistance mechanism of the drug, then I would rather not go for it, but rather go for other kind of, more sensitive, more less likelihood of AMR regions. So there are different aspects of drug discovery and definitely ML can help in drug discovery.
Thank you so much, ma'am. I hope all the
Right.
questions have been answered. We have one more question, ma'am. How can
Sure.
machine learning algorithms be used in finding new biomarkers?
Yeah, so that's the same answer, wherein, as I said, there is a geo data set where we look at gene expression data sets. So, if you all are aware of geo database, please go to that and you will see that, it's well annotated in the sense that researcher has said that for this particular individuals, For example, there would be cases where PCOS women and their gene expression status is mentioned. So we could probably similar experiments by, similar experiments, very important. If there are 5 or 6 studies on similar type of, PCOS women without any interventions, you could look at the gene expression pattern of these different samples and see, is there anything that is coming as similar as a pattern across, or a same pathway that is coming to be enriched in all the different studies. But very careful, you have to, inclusion exclusion criteria has to be very stringently defined. You do not want to club PCOS women, women with PCOS having an intervention or a comorbid condition. No. So if you are using an inclusion and exclusion criteria very carefully, then you could probably compare the expression status from different studies so that your level of dimensions is much more than just one researcher, and then see for common patterns in expression. And then, if one of these could be used as a biomarker, which is not yet known, but that has to be studied again, I mean, that has to be validated again on a population. You would only get possible or putative targets, but then as a biomarker, it needs a different level of validation. And of course, as again I said, you have to benchmark with what it is existing and how this can actually improve.
Are there any questions, students, participants? Okay, so as there are no questions, we now move on to the valedictory function. I, Mr. Abhishek Das, on behalf of Patkar-Varde College and the entire faculty from the Department of Microbiology, Biotechnology, and Bioinformatics, feel privileged to propose the vote of thanks. I would like to extend a very healthy vote of thanks to our speaker, Dr. Susan Thomas, for taking out her precious time for illuminating students and making this webinar a great success. I would like to express a profound gratitude to our management of Chikitsak Samuha, CEO Dr. Mala Farkar ma'am, our principal Dr. Pratibha Gaikwad ma'am, Chief Coordinator and Coordinator Mrs. Ruchita Dalvi ma'am for their moral support and guidance. We are also grateful to our sponsoring organization RUSA. This event wouldn't have been successful without the constant support from our staff members and wouldn't have run smoothly without the technical team. Last but not the least, thanking our wonderful participants who have turned up in such great number from all over the country and for their active participation throughout the webinar. Once again, thanks to all for their cordial operation. A feedback link has been posted in the chat box. It will be open for next half an hour, and e-certificates will be mailed to all the participants in 2 to 3 working days. I declare the session is closed. Thank you.
Thank you.